Hi everyone, I'm Hazel from the SpatialX team at Bioturing, and welcome to the SCSC Club today. So for today's section, we are going to explore the ability to work with Xenium data on SpatialX. So we will go through how you can submit Xenium data on SpatialX and how you can perform some basic analysis on the platform, and if we have some time, I will walk you very quickly through different kind of advanced analysis that you can perform using your Xenium data. And along the way, please feel free to let us know if you have any question. You can put it in the chat or in the Q&A box so that I will try to answer everything. So yes, let's start right away. So in here, just one notice that if you are a Biotrain users and if you're using SpatialX right now, you can actually access the example dataset that we're having here. So let me go to the platform where I can show you in my storage. If you open the Biotrain public, in here, I will open on my end here. Okay, if you open the Biotrain public S3 bucket and then you open the SpatialX folder, and you should be able to see the example Xenium human kidney with protein panel dataset inside, and you can easily, submit the data for trying out the Xenium data on our platform, or you can even visit 10x Genomics to get this example dataset and upload it to our platform. Okay, so I will go quickly through so how you can submit the Xenium data onto our platform. So as you can see in here, our submission workflow is broken into two steps. So the first step will be upload. So the first step including either you uploading your data from your local computer or you link your cloud storage. So it can be either S3 bucket, SFTP. So SFTP is very convenient because you can link directly, let's say from your supercomputer or for your institution computers, or from different kind of local storage to our platform directly. And you can also use the Google Drive as well. And after uh, linking the buckets or uploading the files onto the platform, you can then start the submission process. So let me actually go back to the platform and guide you how you can do that. So if you on the platform, you can click new study in here. Let me make it a little bit bigger. And I have my study team here. And then in order to submit the Xenium data, you can select the platform Xenium, and in here, I have my example data set of human kidney. I open it. And now, for your convenience, you can just smart fill all the required files. So if you have every files and folder stored in one big folder, you can just smart fill into the submission panel. And now you can fill in the name. This can be the kidney one. Okay. Okay. So before we moving on, I want to share with you some specific tip or about the data submission for Xenium. So as you can see in the submission panel, there are two mandatory files. They are the segmentation and the transcript files. And of course, you can click the Smart Fill button to auto pull all the required files to the submission panel. And then, yes, we are talking about spatial data, and Xenium specifically has a lot of different kind of images. And as I listed here, you can submit, let's say, most of the images that you have for your Xenium dataset onto our platform, including the morphology image, which is the DAPI image. And if you have an H&E image, like a pair H&E, you can also submit to the platform. And in here, if you do have some alignment file to align the H&E to the morphology image, then you can submit it along. However, it's okay if you don't have this file because you can perform image alignment after data submission is complete. So just feel free to submit your H&E image here. And this is a new update, the protein and morphology or morphology focus images. So for most of you, if you don't have the protein channels, you will have the set of 4 different channels, the morphology channels, the DAPI, Ekaterin, 18S and the vimentin channels to capture the morphology of the cells. And you can submit these image onto this one. And usually those images will be stored within the morphology focus folder. And once you submit, and even if you run the protein panel directly from Xenium, so yes, right now, if you work with Xenium, you can start running the proteomics panel. And when you submit the morphology focus folder on to our platform, we will automatically combine multi-single channel IF images into a multi-channel IF images. And if I want to show you quickly how it will look like on the platform after submission. So in here, this is the dataset already submitted. And if I open my preferences panel, I can have my morphology image of the DAPI. I will turn off the cell to view the image a little bit better, the DAPI image. Then I have the H&E image, okay? And then on top of everything of that, I also have my kidney protein image. So if I turn on this, I can visualize different channels, and you can even save your channels as different groups like this one, and I will show you a little bit on how you can save the channel groups and how you can, adjust the channel signal right here. Okay, if I go back here, and so after you filling in all the required files and folder, then this one is a little bit like tip for you if you work with multi-slice. So if you work with one section at a time, then this is easy like this one. However, we also support the multi-sample submissions where you can automatically pull one big folder onto this auto-populate sample button, and we will automatically recognize all the subfolders as the samples. So it requires a little bit of files and data organization beforehand. However, after that, the submission will be super fast. So this is how you can organize your data. You have a big data set folder. So this is one example folder. And within this big folder, you will have multiple subfolders, right? And each folder or each subfolder here will contain the data of one samples or one slice, and you name it accordingly. Then within each of the subfolder, you will have all the required files in here, the uh, the segmentation file, the transcript file, and different kind of images. I highlight two of them as in blue because they are mandatory files. The other images, if you have, then you can include. If you don't have, then that is absolutely okay. If you go to the platform, then you can see the auto-populate sample over here. And when you select Xenium, you can be able to, let's say this is one example dataset that I have, which contain a lot of samples. And if I drop it here, it will automatically recognize all the samples as individual folders as of in here. Okay. And if I cancel this one, you can submit. Okay, so that is for the submission. Then what was next? What's next for the submission? So in the submission, you can also perform some filtration, some quality control, where if you click on this QC and pre-processing button, in here you will notice that we have two different set of QC, which basically the same. So what is the difference between them? The QC and pre-processing down here, I click save so it's easier to visualize. This one is a global QC, which means if you have multiple sample like this and you perform QC down here, and it will be automatically applied to all samples. And this one, it will filter your cells based on the number of genes, number of transcript, the percentage of mitogenes, and the percentage of negative genes. Okay. However, let's say that you have different samples, but you want to apply different set of normalization on different samples. Then you can open individual samples and you can customize the QC and processing for the individual data like this. And if you don't click, then the global QC will be applied. And another thing is skip biostring pipeline. So most of the time we recommend user to run the biostring pipeline where we will automatically run the embedding, T-SNE UMAP, and clustering for you as you submit the data. So it's easier, let's say that when you open the dataset firsthand like this, I should turn on the cell and I can adjust this one, this channel, okay, to make, reduce the background noise. So this is, yes, this is how you can adjust the channel, the signal intensities, okay. Right when you open the dataset, you will see the default Leuven clustering appear. So it's very easy for you to, like, visualize the data, and you don't need to perform the clustering anymore. However, it's okay that you want to perform or like redo the clustering or embedding. Then at that time, you can go to the 2D mode up here, and you will see the embedding, Disney, and clustering. So you can rerun. And you will also see the result that we run for you by default. But you can always rerun, or, with different set of parameters. Okay, I go back to my slide here. Okay, so now you have the data. How can you start working with the data? How can you start working with the data? The first thing would be the data visualization. So as I mentioned earlier, using the Xenium data, you can submit both your proteomics and transcriptomics data to the platform. And I also show you how you overlay the images together. And in here, if I go back here, I go back to the image. Okay, I go to the preferences panel. Okay, and I turn on my image up here. I should increase the background noise, increase the threshold to reduce the background noise. Okay, this one. Okay, that is. And if I zoom in really clear, you can see this is a set of perforation and differentiation cells. So yes, and you can easily switch between different images as well. So let's say that right now I'm looking at the immunofluorescence signal, but I can also switch to the H&E image, and to visualize, I can make my fluorescence signal a little bit, reduce opacity, so that I can see not only the H&E, the morphology, but also the protein, the phenotype of the cells at the same time. And I can even see the clustering of the cells at the same time. Okay, now the next thing that you can see, this is like a very quick tip for you, that if you have multiple sample, how can you deal with multiple sample? So if you do have multiple samples on SpatialX, and when you work with Xenium, you can view them in the grid view. Up here, you have a layout button where you can view multiple samples side by side, or 4 samples side by side. And the advantage of the grid view is the fact that you can query genes across multiple samples. However, these samples are separated. So there's no combination here. It's just a comparison between samples. And I will show you a little bit later on how you can combine them together. And beside the grid view, you can also view in mixed display. I will show you on the platform. So now I'm in the image projection. So if I switch to the dapple layout, and then I switch to the mixed mode, like, so yes, this is how you can visualize the EU map and the image distribution at the same time. And it's very easy to, find the macro genes or query genes, with the two projections like this. So if I go back here, I would then go next with a very common question about the visualization. Now you have like a pair H&E image, but you don't have it when you submit the data. Like, you run the pipeline, and you have the Xenium data, but you don't have the H&E. But maybe like a few days later, or like later on, you now finally have an H&E. How can you submit that H&E, and how can you actually overlay the H&E on your data? So if you go to the in here, I will quickly show you how you can do that. Okay, now you go to the preferences panel, and you click on the plus button within the image tab. You click on it, and then you just upload the image. And we do support different kind of a format. So it's very diverse, from like lower quality like PNG to very high quality TIFF file, or even big TIFF file, or SVS and DPI H&E. So you can just upload it, locally or from the cloud storage. And once you upload the image, how can you align the data together? Now, if you hover to your H&E image, and you will see align image button, so where you can actually overlay the image, the H&E, on top of the different kind of images. And not only that, you can even turn on the image setting in here, and turn on the cell centers or even the cell segmentation. And if I go continue, you can start, put the landmark point to align the image. And in here, another quick tip I want to share, that the fact that normally most of you don't have the same tissue sections for H&E. You have zero section instead. And that is still okay with SpatialX, because in here, if I put some of the landmark point pair first, like right there, and then maybe something below here. So okay, you put the first three landmark point pair, and usually I put it around the image to capture the overall shape of the tissue. And then now you will see the alignment preview. So this alignment preview will very helpful in aligning the, zero images or zero section. Why? Because as you add more and more landmark point, you will be able to see the transformation in real time. And if I go deep into this region, for example, I will see this very dense region right here. And I can actually align up down to every individual cell. And now the landmark point number 4, it will appear here, and the transformation will show in real time. So that, and because we are talking about zero section, right here you are looking at non-linear transformation. So this is not linear, but instead this is non-linear. We try to work with linear, but we figure out that non-linear is much better for aligning the zero section. So yes, you can just put more and more section and preview in real time. And for many of you working with multiple monitors, you can even make this preview on one monitor, expand to another window, and having the alignment on another window. So it's suitable for multiple monitors at the same time as well. And if I go continue, I can save this alignment result. And yes, that is how you can, add image and align your images on top with of the Xenium data. Okay, this is just a quick guideline of how you can do it. And this is how you can align the images and place the key point. Okay, now you have multiple proteins, and because we are working with proteomics as well, how you can manage your protein channel. Okay, so if you go back to your protein image up here, I will close this one and I open this one. You will see that first of all, you will be able to edit, to configure the signal range, to reduce the outlier signal or the background noise of the fluorescent signal. You can click on individual channel to switch between channel. You can add channel, and you can add up to 10 channels. You can view 10 channels at a time. And if you want to change the color, you can just click here to change the color of the actual fluorescent signal. So I will turn off the H&E, maybe turn off the cell, so I can visualize my signal a little bit better. And next, you can go to the channel management to actually group channels into multiple channel group like this one. And you can even modify the channel name, just maybe easier for you to work with different protein names. And once you set the channel as different groups, you can view by groups in here. You can quickly switch and open different, different groups to visualize your different groups of cells or different markers for immune cells or for tumor cells. Okay, now I will go back here to my slide, and then another question. Now, if you work with like Xenium data, you will very usually have a lot of sections at the same time. So how to combine data and how to perform the batch correction. So in this example data, I only have one sample. So I will go to my another data set in here. I will try to go quickly. So for this one here, I will open this. And this is one example data set that I have multiple samples, right? So for this multiple sample, you can actually combine them together. So to combine, you go to this drop down here. So this one, in this section, you can actually switch between different samples, and at the same time, you can combine the data. Once you combine the data like this, okay, you can click and you can combine the data coming from similar technologies. And I will give you one tip. Because this is all single cell. That said, you can even combine Xenium data of different gene panels. And I must say that is a very unique thing that we can do. You can actually combine Xenium 5K with the Xenium 490. And yes, that is one possibility that you can do. Okay, oh, I'm sorry. And once you combine, we'll combine the expression matrices together. And for one sample, you can bring one image. And after you combine, the image will line up on the screen like this. And now, what is the difference between grid view and this one? For the grid view, you can only query the genes across different samples. But for combining like this, you can even remove the batch effect between them. So if you go to the 2D view mode, okay, and you go to the let's say this one, the batches information metadata, it will contain all the sample name and sample ID. You will see that all the cells, they blend in very well with each other because we do perform the batch correction. And you can see in the embedding in here. So in the embedding, when you run, or when you combine the data, we automatically run the batch correction using SCVI. If you have more than 1 million cells, we will run using the PCA. It's just to save the running time. But you can always rerun using either PCA or SCVI. And if you turn on the batch correction, then you will be able to run the PCA with Harmony or the batch correction within SCVI. You can even select different batch level metadata to run the batch correction as well. And on the same line, you can rerun the TCU map clustering, and you can perform the cell type prediction across the whole set of data. So you don't need to perform it one by one anymore, but you can perform on the whole set of sections at the same time. And after you have the annotation, so let's say in here, let's say I go back to the Louvain clustering. After I have the annotation, from the Louvain clustering, you can actually, oh, I will first talk a little bit about the cell type prediction. So our cell type prediction is cluster-based cell type prediction using the reference, which is our library of over 2,000 single-cell RNA-seq dataset. So you can access our public libraries of single-cell RNA-seq on Talk to Data. But in here, I will input the Louvain, and then I will predict with default reference. But you can run with the custom reference. You can select some study belong to, contain, maybe this is the lung study. You can select lung tissue, so that it contains relevant cell type. And besides the public reference, you can use your own reference as well. We do have personal reference. If you work with single cell, you produce your own single cell data, you can then transfer the annotation from your single cell to spatial data using this personal reference. Okay, this one here, after you have the prediction, like this one, I will go with the, in here, meta reference prediction of different cell type. Interesting thing is that after you have this, annotation or any information, right, you can go to the metadata management. And if I click on the meta reference prediction, which is the annotation result, I can do this little thing. I can push the metadata back to original sample. So it's a very nice way to organize your metadata where you can perform everything in a combined data. And at the end of the day, you can push it back to original sample so that later on you can visualize them one by one or separately. So it depends on you. Okay, in here, you can just click and push it back. Okay, now I will go back here. I will go through the combined data, batch correction. I have some tip in here, and also how to handle the metadata. So you can take a quick screenshot of how we can handle that. And if there are any tips that, that you can use for your own research. Okay, now this is embedding and combine sample. Now we go next thing. Oh, I'm sorry. I think we have a little bit duplication issue. So I will go next here to the cell type prediction, which I just mentioned a little bit earlier, that you can perform. I will turn on the slideshow. Okay, you can perform using our cell type prediction tool and using maybe our own reference or using your own reference like that. And at the end of the day, because sometimes we do understand that you can still work with Xenium of smaller gene panel, and it can be very limited when it comes to small gene panel. So yes, you can perform the annotation on your own. So how can you do that? So let's say if I go back to my slide in here. Oh, how can I hear? Okay. Now, if I go back to my sample, my data back here, you can actually perform the marker genes, right? You can input, let's say the in here by metadata, you can input the Louvain clustering result there. And then if you run the marker genes, you will have automatically the list of marker genes for all clusters in your dataset, just like that. And later on, you can use these genes to predict, to annotate the cell type based on literature. Let's say if I go back here, I can quickly open my gene set, which I already saved or I already submit earlier for the cancer-associated fibroblasts, for example, like this. Oh, I don't have many of them in my data set. This is a very, common case for the uh, in here for the Xenium when you don't have a genes in your gene panel. And I apply this thing. So you can quickly apply and query some genes like this. You can turn on the AU cell, and then you can select the cells based on the AU cell score. The higher to one, the more enriched level, and you can select the cells based on the AU cell expression, and you can annotate the cells that way. Once you annotate, this is where user or you guys usually feel quite confusion. What is metadata and what is label? So metadata is a specific kind of information. Right now, we are dealing with cell type information. So in here, I will say I will put it a very general name, cell type. So it will be the type of information. And now the label will be the exact name or identity of this cell group of my 12,000 cells. So they maybe they are CD163, they are macrophage. Okay, and this is how you can distinguish between the two. Metadata is the kind of information, cell type, disease, age, condition, sex, whatever, and the label are the identities of different cells group within. And when I annotate, it will show me now I have my macrophage annotated. And of course, I still have a lot of undersized cell. So in the metadata, there will be always information for all the cells that you are dealing with. So for any cells that you don't assign the identity yet, they will remain unassigned. But if you go and continue to annotate cells more and more, then these unassigned cells will be smaller and smaller and smaller. Okay. Now, if you have the macrophage like this, you have the annotation, what can you do next? I will spend I think my 2 minutes to go quickly through everything that you can do. The first thing, of course, the marker genes, and then you can do the DGE analysis as well. So in the DGE analysis, you can select the cells group of different, identities, different metadata, and then you compare the list of up or down regulated genes. The important thing in here that is the metadata creation, because in the DGE you need to select the cells for the two groups, right? So many times you will be quite confusion on like how can I create the metadata. So it's very easy. You can use the lasso in here to select the region that you want to to add an identity. You can annotate to a new metadata, or you can use existing metadata like the Louvain or the sample ID. And if you want to combine some of the information together, you can go to the manage metadata to quickly add in here and combine metadata together to have different layers of information in one single metadata. And at the end of the day, you can go to the differential expression to select the metadata for running the analysis. Okay, I go back here. And after you run the DGE, you can even use this list of DGE to run the pathway enrichment using GC, GZ enrichment, and you can visualize with different kind of figures. Okay, I think that I don't have enough time for many of the advanced analysis that you can use, but good news with Xenium, you can run, I must say, all kind of analysis in SpatialX, including in here. The first thing will be the region segmentation, where you will be able to define the spatial niches between your tissue. So it's clustering, but with spatial context. And if you run, you will also have a set of signature genes for different regions and the associated pathway as well. And I'm coming up next, you can perform the distance query where in here, I'm working with neoplastic cell and I want to find the immediate neighborhood of neoplastic cell surrounding my tumor cells. So I can do that. I can just select a radius, the size of my neighborhood, and it will quickly show me the distribution of the neighborhood. And then you can even run the cell proportion to break down the cell type composition of your tumor microenvironment. And next thing is you can export the figure on BioVinci to further modify the figures, to make it prettier. And yes, you can export almost all kind of figures onto BioVinci. And next thing behind the distance query, you can also perform the neighborhood analysis. So it's a more comprehensive picture of the whole tissue, which cell types are close together, and you can even form the different neighborhood on the tissue in which each neighborhood will have a distinguished cell type composition. So after the neighborhood analysis, to define the different spatial domain on your tissue, you can then answer the question like what kind of cells or like how the cells communicate with each other using the cell-cell communication. You can even confirm these white arrows. They are the interaction, right? Ligand receptor interaction. You can confirm the interaction using the protein signal on the immunofluorescent image, or you can confirm the morphology of the cells based on the H&E image. And at the end of the day, we do have some feature set for pathologists. So if you're a pathologist, you can run the image segmentation to automatically cluster your H&E image. And even in the near future, we will also have the auto labeling for H&E image as well. So yes, that is almost everything that I want to share today. And if you want to find some resources, so if you want to find some resources, you can go to our documentation page, or you can go to our blog, and you can always book an one-on-one session with us to walk through all the features in very detail. And yeah, we're really happy to try to help you guys with how to analyze the data, because we do understand that spatial data sometimes can be it can be a mess. So just please let us know to our support email, and we will try to, meet up and discuss further. And right now we have some little poll, Q&A that you can try to answer on the screen that my colleague will put on the screen. And also, if you do have any question, just let us know so we can. Oh, so in here I have some requests to include detailed video tutorial for the advanced features and analysis. So yes, of course, we will include that part for the advanced features and analysis, because yes, this session is very short and it cannot cover everything. So we will upload that one and we will definitely share it on YouTube. So yes, thank you a lot. In here, let me turn on the pause. Okay. Okay. Yes, I see the pause in here. I'm sorry. So I think we already over the time, but please let us know if you have any questions. You can put it in the chat or in the Q&A. We'll try to answer everything. Okay. Mm. Yeah. Here, let me see if we have any other question here. Okay. Oh, I'm sorry. Before we go, we have one question. The transcript segmentation using SpatialX. Oh, just I'm sorry, I cannot reach out to you, but please reach out to us to support email. We are currently working on the transcript segmentation. The re-segmentation after maybe you perform DAPI, then you can perform the transcript segmentation. So please send us an email if you're interested in this feature, and we can discuss more. Yeah, thank you.